This is a video to help you with Chapter 8.1 on metabolism. Now, Chapter 8 includes three major topics. So that would be enzymes, photosynthesis, and cellular respiration. So we're going to cover this along with the standard level content on Chapter 2.5 on enzymes. So a lot of this will seem kind of like a reiteration. But keep in mind you'll need to know this stuff when we get to photosynthesis and cellular respiration for the higher level content later on. All right, so this crazy looking nonsense is what's known as your metabolism. And I know we often talk about metabolism with like, oh, I have a low metabolism, or I have a slow metabolism. If I even look at a hot dog, I gain 5 pounds. Well, that's not really how we should be using that word. Metabolism is a sum of all the chemical reactions that occur within a living organism. And those reactions, of course, include both anabolic and catabolic reactions. So just to review, anabolic reactions are building molecules. So they're forming bonds through condensation reactions. This requires energy. And some of the ones we're going to look at include photosynthesis. Okay, so taking carbon dioxide molecules, taking out all these carbons, and forming one large glucose molecule. This requires energy in the form of sunlight. There are many other anabolic reactions, however. Catabolic reactions are breaking molecules down. Okay, that involves hydrolysis, and this releases energy. So a great example here is cellular respiration. Taking glucose and breaking it down into its subcomponents, which will eventually get released as carbon dioxide and water, and that process releases energy for the cell to use. Now, these reactions are hardly ever one-step simple reactions. Instead, they happen along what we call a metabolic pathway, or a sequence that shows the intermediate steps of a reaction. So just like at one time, and don't draw this, is just an example, at one time you were a combination of sperm and egg, and then you were an ugly little fetus with gills and a tail and webbed feet, okay? And then you grew into a baby, and then you grew into an adult, okay? So kind of like not really a one-step process. Well, metabolic reactions like photosynthesis, they happen the same way. So here's how we're going to draw them. So let's say we start with substrate A. I'm just going to letter them. You could have numbered them. It doesn't matter. Okay. Substrate A is going to magically turn into substrate B. Sorry, my dog is going nuts out there. Okay, and this is going to be done with, of course, the use of an enzyme. Okay, so our friends' enzymes are back. And this might get turned into our final product or end product through, of course, the use of a different enzyme. Okay. So I know oftentimes we kind of say, oh, well, overall glucose and oxygen gets turned into carbon dioxide, water, and ATP energy. It sounds like this just magically turns into that. Well, it's not that simple, as you're going to find out. There are a bunch of intermediate steps involved, and almost all of them require enzymes. Now, since most of these processes require enzymes, they're going to happen in the spots where the enzymes are located. Hello, that seems to make sense, right? So we know photosynthesis is carried out in the chloroplast of cells. Well, how come? Why isn't it just carried out in the cytoplasm? Well, because the chloroplast is where all of these important enzymes are found. Okay, so just kind of a bit of common sense knowledge there. Okay, this video is becoming impossible to make without my dog. He's being a pain in the rear end and insisting to be a part of things. He's feeling very left out, so we're going to let him join in over here and be his usual pain in the rear end. Okay, while we do that, let's go back and review kind of a couple things that we already know about enzymes. Okay, so enzymes fit into one of the two basic models, right? So the old school model of lock and key. So that enzyme has a fixed active site, the substrate fits in there, and everything proceeds as normal. Well, we know that's not entirely the case. Now we know that enzymes really work on this induced fit model. Okay, so this still talking only one substrate per type of enzyme, but when these enzymes enter and bind with the active site, it changes the shape of the enzyme just a little bit. So it kind of like curls around it just a bit. Again, don't get confused. Lactase can only work on lactose. Amylase can only work on starch. Pepsinogen can only work on proteins, okay? So even though these active sites are kind of changing shape a little bit, they're still specific to their substrates. Now, regarding enzymes, okay, enzymes carry out all kinds of reactions. Some enzymes break things apart, some enzymes help build things, okay, and they're basically involved with two different types of reactions. And we're going to take a look at this one called an endergonic reaction. And I know in your notes I didn't really put a spot for you to write this down, but you may want to do that anyways. All right, endergonic reactions basically look something like this. Okay, so over here we have the progress of the reaction. Okay, so this is kind of like on a time scale almost, moving that way. And over here we have energy. All right, I'm going to do my non-enzyme catalyzed reaction in red. You can do it as a solid line or dotted line, whatever. All right, in an endergonic reaction, the products, so what we end up with, are going to have more energy than the reactants. Now, don't forget, we also have to include the activation energy. Okay, so that's going to look something like this. So here are my reactants. I got to put in my activation energy, and then here are my products. Okay, so we have reactants, we have products. Okay, here again is my activation energy, but overall my products have more energy than the reactants. Well, that's kind of like photosynthesis. Okay, in photosynthesis, your major reactant, carbon dioxide, has a lot less energy than your major product, which is glucose. Okay, so that means that we have to be putting energy into it. That sounds to me a lot like an anabolic reaction. Enzymes, okay, help lower the activation energy. So there's less energy needed to get it started. But we notice we still start and end in the same places. Okay, so it may involve less energy to initially get that reaction started, but we're still going to have to add in energy overall because our products have more energy than our reactants. And that's what we call an endergonic reaction. Gonic referring to energy. Endo or inner, remember that means into, like endocytosis going into the cell. Endogonic having to put energy into a reaction in order to make it work. Quite the opposite is true for exergonic reactions. So again, we have our reaction progress down here on the x-axis and energy here on the y-axis. Okay, and in an exergonic energy, that means this is going to release energy from that reaction, and that's because the products have less energy than the reactants. Something like cellular respiration, where we're going from a high energy reactant to a low energy product. And these are usually catabolic, right? Breaking things down. So here I'm going to start again with no enzyme. Here are my reactants. I put in my activation energy, and then here are my products. So my products have less energy than my reactants, okay? So something like in this situation. And that means, and I'm just going to back up for a second before I draw enzymes, all of this energy here, you don't have to draw this, but the difference between the energy in your reactants and your products, all of this energy gets released for the cell to use. So in the case of cellular respiration, all this energy can then be used for generating ATP, okay, and other cellular processes. So that difference in energy is really quite big. If I add an enzyme into this situation, it's going to decrease the activation energy. But again, I'm still starting and ending at the same places. So it makes it easier for this reaction to take place because I don't have to put as much initial energy into that. But my reactants and products are still the same. And that's an important thing to remember about enzymes. So just a quick review from chapter 2.5. How do they do this? Well, what they do is they lower activation energy by making sure that molecules are in favorable positions and they weaken bonds in order to make that reaction take place without having to add in so much energy or energy of activation. So let's break down how these enzymes really work. Okay, so the first thing that's going to happen is the surface of the substrate, so whatever reactant is in our equation here, is going to make contact with the surface of the active site of our enzyme. So that's going to touch this. Then the enzyme is going to change its shape to accommodate the substrate. Again, that's our induced fit model. It can still only fit this certain substrate, but it kind of conforms to the shape of that substrate only after the substrate binds there. And this is going to cause the formation of a temporary complex called the enzyme substrate complex, or ES complex, but I'd really rather you write this out for the first time. And that's just a temporary state where the enzyme and the substrate are stuck together. Then the next step really kind of involves our reaction, right? Once the enzyme interacts with the substrate and forms that complex, the activation energy is lowered and the substrate is altered, okay? So what does that mean? Well, it means that either the two substrates that were separate are now together, or if it was something that was together, now they're broken apart, whatever, okay? The substrate has been changed. The reaction has taken place. Okay, then the products get released from that active site. And notice that that enzyme is unchanged and it's free to work on other substrate molecules. So it's going to go something like this. The enzyme and the substrate form an enzyme substrate complex. The enzyme performs whatever reaction is taking place and turns that into a product. That product gets released and now this enzyme is free to start that cycle all over again with a substrate, okay? So if I have 1 billion molecules of lactose, one molecule of lactase enzyme will work its magic. It'll release the glucose and galactose, and then that unharmed or unchanged lactase molecule is free to work on the other, I shouldn't have made such a big number, 999,999,999 molecules of lactose that are left over. Now, this all sounds well and good, but what we don't want is a bunch of enzymes out of control, doing things they shouldn't be. So part of our natural processes involve this concept of inhibition. And when I always hear this word, I always think of like how my biology teacher in school was like, now everybody, the use of alcohol can inhibit your decision-making processes. Okay, and that means stop, right? Okay, so alcohol kind of impairs or decreases your ability to make good decisions. Well, the same thing can happen with enzymes, okay? We can get an inhibitor, something that decreases the effects of an enzyme. Now, this can be good, like maybe we don't need this enzyme to work anymore, or it can be bad, like maybe, yikes, some kind of pathogen is causing some kind of problems, or toxin is causing some kind of problems that's interfering with our enzymes' ability to do their job. The first one we're going to take a look at is something called competitive inhibition. It's exactly what it sounds like, thank God. And this is when a molecule that is shaped similarly to the substrate kind of hangs out here in the active site, and it blocks that substrate from joining with the enzyme. So this is what's supposed to happen. Let's say this enzyme is supposed to take this substrate and split it into two. Well, in order for that to happen, this substrate has to cozy up here in the active site. If I add in a bunch of molecules that look like that substrate, they might be able to fit in here. Now, they're not the substrate, so the enzyme can't do anything to them, but it's still blocking this active site, and it's preventing this normal reactant from being able to bind here. All right, so that's going to look kind of like something like this. So let's say I have an enzyme with an active site that looks like this. Okay, and here is my, well, that was dumb. Here's my enzyme. Okay, and I have a substrate that looks like this. Okay, so here is my substrate. And you can see how this substrate might fit into the active site of the enzyme. Well, what's happening is that some kind of competitive inhibitor that is shaped like the substrate but isn't really the substrate is sitting in there and blocking. Let me make this even more obvious that it is not what it is supposed to be. Okay, it's sitting in there and blocking that active site, and the substrate can't bind there. So this is some kind of inhibitor. And now this can't get into that active site. Okay, so something like that. Wow, substrate, sorry. So one of the examples that you should probably know for this one has to do with antibiotics. So when we take a medicine that is supposed to destroy a bacteria, we have to remember that that bacteria is a living cell. So if we take a medicine that destroys all living cells, we've kind of just screwed ourselves, because that medicine is going to mess with our own cells. It's going to kill our own cells, and we don't want it to do that. We just want it to kill the bacteria. So what some clever folks have found is a way to inhibit some of the enzymes that bacteria need. So for example, in order to build their cell wall, they have to produce something called folic acid. Folic acid requires an enzyme. So these drugs called sulfonylamides, say that 10 times fast, what they do is they block, okay, this active site. So here's the enzyme that produces folic acid, okay, here's the substrate that is supposed to fit in there and then become folic acid. Instead, these sulfa drugs sit in and block this active site. Notice how similar they look. That is so shady. It's not the actual thing, they're different, but it's like putting a fake key into a lock that kind of fits, but it doesn't turn it, okay? And so what that does is it inhibits the bacteria's ability to form a cell wall, and then those bacteria die. So again, we don't build cell walls, we don't use this enzyme, so it doesn't matter to us, and that's one of the great parts about using specific enzyme inhibitors as medicine. Okay, the next type that we're going to talk about is non-competitive inhibition. Non-competitive is kind of what we call it nowadays. The old school term is allosteric inhibition. You still need to know that, okay? Allosteric sites, allosteric inhibitors, you got to know that those are non-competitive. Unlike the screaming children right outside my door that are ruining my video, but I digress. Okay, so this is something that binds to the enzyme, but in a place that's not the active site, okay? When it binds to that allosteric site, we call it, it changes the overall shape of the enzyme, including the active site, making it non-functional. Like competitive inhibition, it could be reversible. It could also be permanent. All right, so that's looking something like this. Again, here's our enzyme that's supposed to take this substrate and split it into two. Let's say some kind of an inhibitor binds to what we call the allosteric site. Again, notice that is not on the active site. It's somewhere else on the enzyme. When it binds there, it changes the shape of the active site, and now this substrate can't fit in there. Okay, womp, womp, womp. So let's draw our enzyme, and I'm going to draw this twice. So I'm going to draw it before we have an allosteric reaction. Okay, so here it is. You can tell that it is supposed to fit this substrate. Or at least I hope you can tell. Yikes. Okay. Now, if we have an allosteric inhibitor, what's going to happen is that on my enzyme, holy crap, I hope you're better at drawing this than I am. If not, you should just quit life now and become, I don't know, some kind of wandering traveler asking people for money. Anywho, okay, let's say I have some kind of inhibitor bind to an allosteric site. What that's going to do is it might change the active site shape, and now my substrate can't fit in there. Okay, so now this active site won't fit this enzyme. All right, so maybe we should label some of these things because we love to watch Mrs. Hawes write these things. So this is some kind of inhibitor. Okay, this is what we call the allosteric site. Here is our, I want to call this our changed active site because it's still an active site. It's just no good anymore. And here is our substrate. One of the classic examples of non-competitive inhibition is mercury. Mercury is so nasty. It's naughty. It's a reason why it's actually illegal for teachers to have mercury thermometers in their classrooms anymore because it does bad things. It binds really easily to proteins, and yikes, enzymes are made of proteins. It can bind to lots of different types of proteins, and again, that causes the change in shape, making them unable to catalyze their reactions. So that could lead to blood clots, tooth decay, gum problems, birth defects, lots of different things. It's one of the reasons why pregnant women aren't supposed to eat things like tuna. And here's why. This is actually really fascinating, so don't fast forward. Okay, let's say little fish, that's a fish, just pretend like you know that. Okay, let's say they have mercury in them. Okay, let's say that a bunch of these little fish, okay, I'm really struggling here. A bunch of these little fish, okay, get eaten by a big fish. Well, that big fish can kind of digest all their body parts, but the mercury accumulates. It doesn't go out of their digestive system. And why not? Because these are some sad fishes, I'm just saying. That's because the mercury is binding to those proteins and it's not going anywhere. So now this big fish has all of the mercury that was in the little fish. Okay, now a fish that's big is going to eat thousands of little fish in its lifetime. So it's going to accumulate all of the mercury that's in there. It's called bioaccumulation. Pregnant women aren't supposed to eat fish like tuna because tuna are big fish and could have high levels of mercury in their tissues. And that could cause some serious problems with a lot of the proteins and enzymes that go along with fetal development. All right, and the last type of inhibition that we're going to talk about is kind of a good kind, right? So I know the first two we talked about the kind of like bad examples of how to make things go wrong with enzymes, but this is actually a good thing. So remember our discussion on positive and negative feedback loops, okay? If something is a positive feedback loop, that means as one thing grows, it causes a growth in another thing, which in turn causes a growth in the first thing that you were looking at to begin with, okay? So that's very different than negative feedback loops. Negative feedback loops bring things back towards middle values. These guys are runaway loops. These things bring back towards a central value. Okay, so let's say if your body temperature rises, you sweat, that means it brings it back to normal temperature. Okay, so end product inhibition is an example of one of these negative feedback loops. Oh my gosh, it reminds me of a terrible, like horrible, really mean IB exam question I saw one time that said, describe 2 examples of negative feedback loops in cells. And people were like, holy crap, I don't know. And they wrote all kinds of junk on there. Well, here's one you can talk about. Okay, you can talk about end product inhibition of enzymes and sound really smart. This prevents a cell from making more of something when it already has a sufficient amount. Okay, so let's say we're looking at this metabolic pathway. Our initial substrate gets turned into an intermediate via an enzyme, another intermediate via another enzyme, and then our end product via a final enzyme. This is going to continue to happen as long as there are enough substrates. And so we may end up with a whole crap load of whatever this end product is when we really don't need any more of that. Thank goodness most of these involved end product inhibition. So what's going to happen is something like this, okay? During that pathway, when we get to the point where the end product is produced, that end product will act as an allosteric inhibitor to one of our first enzymes. So it'll actually bind to our first enzyme, meaning that the original substrate isn't going to be able to bind there. If this can't bind to the enzyme, then this whole reaction pathway shuts down. Okay? So as we increase the number of substrates, okay, if we already have a bunch, then it's going to be inhibiting a bunch of these enzymes. As these substrates start to dwindle or disappear or get used up for cellular processes, then these enzymes aren't going to have these inhibitors anymore, and they can continue on through the metabolic pathway. So if you're looking for a diagram to draw, there's no way I'm attempting my terrible drawings for this complicated process. Take a moment to draw maybe something like this or like this. All right, and for an example of this, we're going to go back and talk about bacteria again, specifically E. coli bacteria. And they need 5 different enzymes, okay, to take the amino acid threonine and turn it into isoleucine. Well, if they already have enough isoleucine, what's going to happen is this isoleucine is going to bond to the allosteric site of the first enzyme, and then none of this happens. We can't jump forward. Okay, these have to happen in sequence, and if one enzyme has been inhibited, then this reaction pathway stops. So you may be wondering, how in the world do we know all of this crap? Well, some of it was done through some pretty gnarly experimentation, but a lot of those experiments are really time consuming, and there's just no way that we can carry them out with enough trials to really be able to pick up on some of these super intricate patterns. So what we've done, I have no idea how to do this, I just know how to talk about it. What scientists have done is they've been able to use computers to run simulated experiments, like millions of them. That would take humans like years and years and years and years, even if they were working on teams of thousands of scientists at a time, to create that much data. And these computers can do it in a matter of minutes. So they can generate all this fancy data, and then they can look for emerging patterns. That should say patterns. That's lame. Okay, there's an N in there somewhere. Okay, so once they find these patterns, then they can start to figure out how these processes work. Okay, so kind of like one of those cool emerging fields of science for any of you looking for a career in something that involves computers. There's a niche for you in the field of bioinformatics. And that'll do it for your first higher level chapter, probably on chapter 8.1 Enzymes.